Hello everyone. In today's video, we're going to be solving AQA GCSE 8461/1F Biology question paper June 2018. Hello everyone. Today we're going to be solving AQA GCSE Biology Foundation Tier paper 1F. In this particular video, we are going to be solving the entire question paper. Today we're solving June 2018 paper. Question number 1 is about cell cycle. Chromosomes are copied during the cell cycle. Where are the chromosomes found? We know that chromosomes are found within the nucleus. So nucleus will be the correct answer. Even though cytoplasm is housing the nucleus, alright, we cannot choose cytoplasm in this case. And ribosomes, they are the ones where we do the protein synthesis. So ribosomes cannot be used as an answer. And vacuole are the ones where we pack the protein after we produce it in the rough endoplasmic reticulum. So nucleus will be the answer. What is the name of section of chromosome that controls a characteristic? Since we are going to control one single characteristic is controlled by a gene, so we will call it gene. Figure 1 shows information about cell cycle. We can see the part of the cell growth, the copying of the chromosome, and then mitosis taking place. Which stage of the cell cycle in figure 1 takes the most time? Obviously, from the pie chart, we can see the copying of the chromosome takes the most amount of time because it's represented in time. So we can see copying of the chromosome requires the most amount of time. During mitosis, cells need extra energy. Which cell structure provides most of the energy? The cell structure that provides the most of the energy at any given time within a particular cell will be mitochondria. Ribosomes, as we already discussed, it is needed for protein synthesis. Cytoplasm is the place where we can find ribosome, nucleus, and all the other organelles. And chromosomes contain the genetic material. So mitochondria is the correct answer. The cell cycle in figure 1 takes 2 hours in total. The cell growth stage takes 45 minutes. Calculate the time taken for the mitosis. So to answer this particular question, because we already know that the cell growth takes 45 minutes, so we will go back to our pie chart, and the cell growth is divided into 1, 2, 3, and like for example, it's covering almost 3 and 3.75. So 3.75 marks is representing that particular time, which is 45 minutes. And if we look into the copying of the chromosome, we can negate this particular portion with this particular portion. So 1, 2, 3, 4, 5. So to find out this particular timing, what we're going to do is we are going to 3.75 of that particular those dashes is equals to 45 minutes. So we will consider that 3.75 x is equals to 45 minutes. Then x is equals to 45 divided by 3.75. And since our for the mitosis portion, it takes about 1.25. So we will then multiply it. 1.25 multiplied by 45 divided by 3.75. We will get 45 divided by 3, which is equals to 15. So our answer will be 15 minutes. Guys, you may also want to use for example, a protector. All right, and by using the protector, you can actually find out the degree, the angle, and once you find out the angle, you can do the same calculation using the angle as well. Figure 2 shows some cells in different stages of the cell cycle. So we can see in A we can see the chromosomes. All right, in B we see the chromosomes are in two different poles. All right, they're pulled apart. And we can see in E we can see the entire nucleus with the nucleolus present. In C we can see the nucleus, but the nucleolus is not visible in C. And in D we can see all the chromosomes are at the equator. They're in the middle. Which cell is not dividing by mitosis? From A, B, C, D, you have to choose which cell is not dividing by mitosis. Now we can clearly tell that B is dividing by mitosis because this is representing anaphase of mitosis, because the chromosomes are pulled to two different poles. We can clearly see, all right, from, for example, D is in metaphase because it is, we can find the chromosomes in the equator. All right, after that we can see A we can see the chromosomes are kind of visible. The nucleolus is not visible, but the chromosomes are kind of visible because you can see that they're pretty large strands here, all right, which are chromosomes. So definitely A must be in stages like prophase, all right, where the chromosomes are just condensing. So C could easily be the one where it is not dividing at all. So we got the answer C because we rejected A, B, and D because they are in stages of mitosis. Cell E in figure 2 contains 8 chromosomes. E divides by mitosis. How many chromosomes will each new cell contain? If we divide by mitosis is a division where the number of chromosomes stays the same. So it will be 8. Why is mitosis important in living organism? Well, mitosis allow living organism to produce new cells. New cells, those that were identical to the parent cell that it came from. So that can easily lead to repairing of tissues. So repair of tissues is going to be the answer. Now, to release energy. To release more energy, the cell will make more mitochondria. Mitochondria are the powerhouse of the cells, so the cell will make more mitochondria. No need to divide. To produce variation, meiosis is needed, and that is a form of sexual reproduction, because in that case, it will create new variation, or it can happen by mutation. However, mutation is not something that a cell can control. Mutation can happen randomly. Okay, to produce gametes, again, meiosis is needed, so that will also be wrong. Plants are made up of cells, tissues, and organs. We have to draw one line from each level of organization to the correct plant part. Now, when it says organ, we know that leaf consists of many different types of tissue that are stacked up together to produce an organ. When many different types of tissues are put together, they produce an organ. All right. Then the next thing says tissue. When it talks about tissue, you see, root hair is a cell, so we have to eliminate that. Vacuole is an organelle, so we have to eliminate that. Xylem cell is also a cell. It's talking about a cell directly. So spongy mesophyll is a layer of cell which are all similar. All right, they have air spaces between them. So this will be referred to as tissue. Tissue by definition has to be similar types of cell arranged together. Figure 3 shows plant cell drawn to scale. Okay, we can see the chloroplast in length. We can see the length of the entire plant cell to be 50 micrometers. So it is going to be 50. Where in plant would the cell in figure 3 be found? So because we are looking into a cell that contains chlorophyll, and the cell is cylindrical in shape, as we can see from the diagram. So definitely this must be found in the just below the upper epidermis. All right, we will find this particular cell in the palisade mesophyll cell, because they are arranged like a stacks of palisade. So we will find it in the palisade mesophyll cell. The next other point says in the epidermis. We know that epidermal cell does not contain any chloroplast. Phloem does not contain any chloroplast. Neither xylem contains any chloroplast. Actually, xylem is a dead cell. All right. Phloem is a living cell, but it does not contain chloroplast. It is mainly needed for the transport of substances like amino acid and glucoses. Next question. Calculate the length of the chloroplast labeled in figure 3. To calculate the length of the chloroplast labeled in figure 3, we will need to first of all do something. We need to measure the length from here to here, which is in this case, let us, we will assume in this case whatever the length for that particular thing is. So for example, let's say if this is for example, if this is 8 centimeters, okay, all right, then we will find out 1 centimeter is equals to how many micrometer, and the length of the chloroplast, let's assume the length of the chloroplast is 1 centimeter. So we will find out, as an example, 50 divided by 8 multiplied by 1, which will give us the length in micrometers. So the answer is 6.25. All right, we can average out this particular answer. We can write equivalent to 6.3, correct up to 1 decimal place. So the answer will be 6.3 meters. Cells in plant root do not photosynthesize. Give one reason why. To answer this question, we have to understand that plant root cells are under the ground. They do not have any access to light, and thereby they cannot photosynthesize without the light. That's why they do not have any chloroplast. As a plant grows, new root hair cells are formed from the unspecialized cells. How does an unspecialized cell become a new root hair cell? An unspecialized cell can become a root hair cell by a process of specialization, which in this case we can call differentiation. Metabolism is the process of breaking down food, or process of making food. Transpiration is the process of water evaporating from the surface of the leaf, and transport is the transport of material from one place to another. So all of these does not talk about how a new root hair cell can produce, which can only form by specialization, which we can call differentiation. Scientists can clone plants using tissue culture. Figure 4 shows the process of tissue culture. We can see the parent plant, we can see white flower within the parent plant, and then what we're doing is we're using a scalpel to remove part of a leaf. Then we're putting that part of the leaf in a growth medium where the cells can separate out and they can individually grow, and then we can see those plants are growing to produce the same white flower. flower. So technically what we have done from this particular plant to this new plants is that we have cloned the plant. Why might scientists want to clone plants? We want to clone the plant mostly because that the particular characteristics that we found in the parent plant, we want to get it back in the new plant, or we want to protect an endangered species because that particular endangered species is no longer producing for example seeds where we can grow the plant from, or maybe the numbers of the plant is way too low. So what we can do, multiply the plant faster, we can do tissue culture on that particular plant, we can clone it to increase the number faster. So to create a new species of plant, as we already talked about, we can only create the same plant back again. To introduce variation into plants, again cloning plant is about producing the same type of plant back again, so we are not producing any variation. To protect endangered plant from extinction, yes, we can clone to protect a plant from going extinct. To reduce disease resistance, cloning does not give any extra benefit to that particular plant because they are of the same genetics. What is the advantage of cloning plants using tissue culture? So it says no special equipment is needed. Now we know for this particular thing is that doing a tissue culture, you will require very sensitive apparatus where you do not introduce any germs from outside. That means special equipments are needed. Plants can be produced quickly. Yes, we can produce the new plants from the existing cells of other plants. So it's very fast because seed production requires a plant to get matured. Once the plant gets matured, then the plant will do flowering, there will be pollination, then only it can go to seed. But using tissue culture, we can just remove any small buds from the meristem and we can just clone it and we can produce many more plants. The flowers are all different color. No, cloning produces same type of plant, so the flower will be same color. The offsprings are all genetically different. Cloning produces genetically identical offsprings. The growth medium in figure 4 helps the plant to grow. Name one substance in the growth medium. So the growth medium must provide the plant as a source of energy. So the growth medium should contain things like glucose or amino acids and also need to contain hormones along with mineral ions and vitamins for the proper plant growth. Figure 5 shows the human digestive system. We can see part label A, B and C. We have to label it. So first of all, in part label C, we can see it's the liver. In part label A, alright, we have the stomach. And in part label B, we have the small intestine. Complete the sentences. Choose the answer from the box. So, digestion is the process of breaking down large food molecules into smaller molecules that can be easily absorbed into the body. So, soluble substances are absorbed into the body. So, we will write soluble. Enzymes help to break down food because they facilitate a chemical reaction. So, we will say that they catalyze a chemical reaction. If the temperature of an enzyme gets too high, the enzyme will get destroyed. So, in this case, we can call it denatured. Protease is an enzyme. Protease breaks down protein. What is protein break down into? So when we break down any protein, proteins are made from amino acids. So we will get amino acids out of it. So amino acids will be the answer. Fatty acid, we can get fatty acid from lipids. We can get glucose from any carbohydrate like starch. We can get glycerol. Also, lipids when broken down produces glycerol. So amino acid will be the correct answer. Why is protein needed by the body? Now you see, body also needs to make its own proteins which are mainly needed for growth. That's why proteins are needed by the body from external sources, because our body is unable to make proteins. So we write the answer like this: body requires protein for growth, for repair or replacement of cells, tissues and organs. So you write any one of those points. Which organ in the human digestive system produces protease? Now, producing protease is a type of enzyme, alright? Now, this enzyme is released by the stomach, alright? Large intestine absorbs water. Liver actually, absorbs glucose from the blood, alright? And liver also is in the process of detoxification. Gallbladder produces bile. So, in this case, stomach will be the answer that releases digestive enzyme protease. In this case, stomach actually releases pepsin, so we can write the name down for future reference. Describe how you would test a sample of food to show that it contains protein. Give the reason for any safety precautions you would take. To test a particular food sample for the presence of protein, because food samples can come in chunks, so we need to ground up the food. Once we ground up the food, then we can add a biurate reagent, which is a combination of copper sulfate and sodium hydroxide, to the food sample. Then the protein, if there is protein present, the solution, which was blue in color, it will turn to purple, indicating that there is a presence of protein. Now, as a safety precautions, because sodium hydroxide is caustic and copper sulfate can actually, if it falls in your eyes, it can cause some burn, right? It can cause some burning sensation. And copper sulfate, if it spills in your clothes, you may get stains, permanent stains. So to protect your eye, you can wear goggles, and if there is any spills in the surrounding, must be cleaned up immediately, all right, because biodate reagent by itself is an irritant and corrosive. Choose the answers from the box to complete the sentence. Obesity can be caused by a diet that is high in fat. If the diet is high in fat, then it can lead to excess deposition of energy within the body in the form of fat. Choose the answers from the box. Okay, obesity is a risk factor for type 2 diabetes, because type 2 diabetes is a form of diabetes where it is induced by life choices that we make. Guys, skin cancer can be caused by, for example, ultraviolet light, and type 1 diabetes is a genetic condition, so that's why the answer cannot be chosen. Draw one line from each blood component to its function. For example, we have the platelet. What do the platelet do? Guys, we know that platelet is needed for the clotting of blood, so the option helps for the blood to clot. Now, the next one, red blood cell. Red blood cell is needed for the transport of oxygen around the body, so this will be that. Now, white blood cell. White blood cell's main function is to kill microorganisms which are going to harm us. So, white blood cell destroys microorganisms. By the way, white blood cell, when it comes in contact, it will kill any microorganism. Alright. So, the other options that were present was transport of glucose around the body. Now, transporting glucose around the body is the function of the blood as in total. Okay. So, we cannot give it as a function for red blood cell, and transports urea. Also, blood is responsible for transporting urea all around the body. Because it shows cross section of the three main types of blood vessels found in human body. Each blood vessel is drawn to the scale, so we can see A, B, C. Now A is representing, definitely, because we have thinner elastic tissue, thinner muscles in A, so A is definitely a vein. We can also understand a vein by the wider lumen. Now the next one, which is a thicker elastic tissue and muscle tissue with a smaller lumen, because it is also at a higher magnification, as we can see. All right, so it's much more smaller than this one. All right, so this one will be a artery. Arteries have thick muscle tissue and elastic tissue. We can see only one single cell layer thickness and with a 7500 times magnification. Even though this lumen size looks bigger, this one must be from a capillary, because capillary is only one cell thick. Which blood vessel has the smallest diameter? So definitely is going to be C, which is the capillary, because it was magnified the most amount of time. Which blood vessel in figure 6 is an artery? So that's going to be definitely B. B is an artery, and reason, we will say that it contains thick muscle and elastic tissue with smaller lumen. Table 1 gives the information about the blood flow in two people. Person A does not have the coronary heart disease. So we can see the blood flow is 250 cm per minute, and person B has coronary heart disease. We can see the blood flow has been reduced in the case for person B. Calculate the difference in blood flow between person A and person B. So what we are going to do is we are going to write 250 minus 155, which is equals to 95. So difference is 95 cm³ per minute. Now the next question says, suggest why blood flow through the coronary artery is lower in people with coronary heart disease. Now we know that person with coronary heart disease, they have a narrower lumen in their arteries because it got clogged or blocked by fat deposits. Calculate the volume of blood flowing through the coronary arteries of person A in 1 hour. The answer must be provided in dm cube. Now guys, about the cm cube and dm cube conversion. 1 dm cube literally means 1000 cm cube. Since 1 dm cube literally means 1000 cm cube, so in this case we need the particular person's blood flow per hour. Now we already know the blood flow per cm cube per minute. Alright, so now we will calculate per hour. To do that, we have to, for person A, the blood flow was 250 cm cube per minute to that particular artery. So we will multiply 250 with 60 minutes, within an hour, and this will give us 15,000 cm cube. Now, since we know that 1 dm cube is equals to 1000 cm cube, so we can divide this 15,000 cm cube with 1000, and our answer is going to be dm cube. Coronary heart disease can be treated by inserting a stent using a coronary artery bypass graft or CABG. Table 2 gives information about each method. We can see the procedure with the stent. The patient is awake during the procedure. Alright, a small cut is made in the skin. A wire mesh is inserted into the coronary artery via a blood vessel in the arm or the leg. And when the procedure is recommended, only when there is blockage present. Alright, only one of the block is present. Then, how long does it take after the procedure? 2 to 3 hours. Recovery time can be up to 7 days. And risk for heart attack during the procedure would be 1%. Chance of failure within 1 year would be 40%. However, in the case of CABG, alright, so coronary artery bypass graft, the patient is not awake during the procedure, means general anesthesia must be provided. The chest is cut open to access the heart. A section of blood vessel from an arm or leg, specifically a vein, it is used to create a new channel for the blood to bypass the block in the coronary artery. When multiple blockages are present, then it should be done. When multiple blockages are present within that same artery, then it should be done. And in the case of CABG, sometimes a triple bypass or a double bypass needs to be done. Recovery time at least 7 days within the hospital. Alright, and full recovery will be done within 12 weeks. The risk of having a heart attack during the surgery is 2%, and chances of failure is 5%, which we can see very, very low. Give 2 advantage of using a stent instead of CABG. So we can first of all definitely say that using a stent, person need to stay. So for the advantage using a CABG, give 2 advantages of using stent instead of CABG. So we must provide the advantages of using the stent. Now for the using a stent, the person does not need to stay for long period of time after the procedure and leave the hospital on the same day. Alright, and in the second point, we can say that it is not as invasive as that of the CABG procedure where a major operation is needed. Alright, then the person also has a shorter recovery time after the surgery. After the procedure, the person has a very short recovery time. Alright, so the person can get back to normal lifestyle much quicker. Alright, and then there is a lower risk of heart attack during the procedure, which is only 1%. Now the next question says that give 2 advantages of using a CABG instead of a stent. Now a CABG has a lower chance of failure within 1 year, where in the case for stent it's 40%, whereas in the case for CABG it's only 5%. And the patient only need one operation to treat multiple blockages. Alright, and multiple blockages can also be treated at the same time, which is a very big advantage for CABG and cannot be done using a stent. Aphids are small insects that can carry pathogens with them. Figure 7 shows a aphid feeding from a plant stem. It has a sharp mouthpiece which can penetrate the plant stem, and thereby it can feed on the phloem juices of the plant. An aphid feed by inserting a sharp mouthpiece into the stem of a plant. After feeding, the mouthpiece of an aphid contains a high concentration of dissolved sugars. Which part of the plant was the aphid feeding? Now we know that dissolved sugars are carried by the phloem. phloem. So phloem will be the right answer. Palisade layer, well, stem doesn't contain a palisade layer, first of all. The second one is stem usually does not contain stomata, and xylem can only carry mineral ions in water. So phloem is the right answer. What is the process that transport dissolved sugars around the plant? The process of transporting dissolved sugar will be translocation. Now, filtration is the process of, we use filtration. How do we use it? We use a filter paper to filter, right? So that's not the process. Respiration is the process of breaking down glucose and using it for energy, producing energy. And transpiration is the process of transporting, salt ions, mineral ions, and water through the xylem. So translocation is the right answer. Plants infected with aphid have stunted growth. Explain one way the removal of dissolved sugar from the stem of the plant causes stunted growth. A plant that is affected by aphid, its dissolved sugar is getting eaten by the aphid. So the less sugar will be available for the plant for respiration and releasing energy. And also along with that, we can write other points that are that the plant will have less amino acid because the phloem also carries amino acid. So the plant can produce less amount of protein which is needed for growth. A third answer that can also be written is that since the aphid removes dissolved sugar, the plant has less amount of sugar for cellulose cell wall. So the cell walls are weaker and the plant cannot grow any further. Most aphids do not have wings when they hatch after several generations. Some aphids hatch which have wings and can fly. Explain the advantage to the aphid of being able to fly. Since aphids, those that are able to fly, they can then take off from one plant and fly to another plant, which will allow them to get more food. The leaves of some plants release oils into their surface. Suggest how the production of oil on the surface of a leaf may protect the plant from aphids. The oil can prevent the aphids from attaching to the leaf. So the aphid is unable to attach to the leaf, and the oil may also kill the aphid. Figure 8 shows part of a rose plant. Give one adaptation shown in figure 8 that helps the rose plant defend itself. We can see in the diagram that the rose plant have thorns in them. So the thorns will prevent it from getting eaten by herbivorous animals. Figure 9 shows a plan of a garden containing rose plants. We can see the direction of the wind flowing, and we can see the rose plants. Now we can see A, B, C, D, E, 5 rose plant labeled. Plant A has the fungal disease rose black. Which plant in figure 9 is the fungus likely to spread to? First, give a reason for your answer. Plant A contains the fungal disease, and the wind is flowing in this direction, so definitely it is likely to infect C. So the answer will be plant C, and the reason will be the fungal spore is blown by the wind in the direction of the wind to the closest plant to A, which is plant C. So just one way the gardener could reduce the spread of rose black spot to the other plants in the garden. So the gardener can spread rose bushes out more farther up. This is one way, even if there was a wind, the wind wouldn't affect the fungal spore, alright, and spread it all over the garden. However, another better way that the gardener can deal with it is that the gardener could remove any infect part of the plant, or the gardener could use a fungicide to get rid of the fungus. Earthworms are small animals that live in soil. Earthworms have no specialized gas exchange system and absorb oxygen through their skin. What is the name of the process in which oxygen enters the skin cell? Now, oxygen entering the skin cell will be definitely diffusion, because diffusion is the movement of substances from a region of high concentration to a region of low concentration. Osmosis will be the movement of water from a region of high water potential to low water potential. Respiration is the use of oxygen in breaking down food molecules and releasing energy, and active transport is the process of moving substances against concentration gradient, which in this case the oxygen is moving down the concentration gradient. Table 3 shows information about 4 skin cells of an earthworm. We can see cell A. Outside the cell, percentage of oxygen is 9, inside the cell is 8, and in that way ABCD information is given. Let's see the question. Which cell has the smallest difference in percentage of oxygen between the outside and the inside of the cell. Now you can see in A it's 9 and 8, in B 12 and 8, so definitely not B. C 12 and 10, the difference is 2, okay, the difference is 4 here, the difference is 1, and in the last one the difference is again the difference is 4. Okay, and the question says which cell has the smallest difference in percentage of oxygen between outside and the inside of the cell. So in this case the answer is going to be, because it has only a difference of 1, which cell will oxygen move into the fastest. Now obviously the cell that has the highest difference between outside and inside, then it will have the fastest movement of oxygen. However, the difference needs to be higher outside. We can see in B and in D the difference is the greatest. However, in D the outside has less amount of oxygen, so inside the cell there is more amount of oxygen, so it will be the opposite way around. Oxygen will move out from this cell. However, in the case for B, the outside has higher concentration of oxygen and the inside has a lower concentration of oxygen, so oxygen will move in the B, it will move the fastest. Earthworms have large surface area to volume ratio. Suggest why a large surface area to volume ratio is an advantage to an earthworm. The large surface area allows an earthworm to absorb more oxygen in a given amount of time, so more gas exchange can occur. The earthworm uses enzyme to digest dead plants. Many plants contain fats or oils. Which type of enzyme would digest fats? So fats are lipids, and lipids are generally digested in the body by using the enzyme lipase. Earthworms move through the soil. This movement brings air into the soil. Dead plants decay faster in soil containing earthworms compared with soil containing no earthworms. Explain why. When there are more oxygen in the soil due to the earthworms' presence, the bacteria living in that particular environment can do more aerobic respiration. Bacteria and fungi are mainly decomposers, so when they can do more aerobic respiration, they can decompose plant material faster. When earthworms reproduce, a sperm cell from one earthworm fuses with an egg cell from a different earthworm. Name the process when an egg cell and a sperm cell fuse together. The process of an egg cell and a sperm cell fusing together is known as fertilization. Guys, remember that do not write sexual reproduction in this particular answer because process name is fertilization. It is, however, a sexual reproduction, but the process of an egg cell fusing with a sperm cell is fertilization only. Some types of worm reproduce by a process called fragmentation. In fragmentation, the worm separates into two or more parts. Each part grows into a new one. What type of reproduction is fragmentation? So fragmentation is a process where the same organism is producing many different or more organisms. Means we can say that this is a form of asexual reproduction because there was no fusion of gametes such as egg cell and a sperm cell was involved. We can also say that this was cloning, however, asexual reproduction is a better answer. Eating food containing Salmonella bacteria can cause illness. Two symptoms of infection by Salmonella are vomiting and diarrhea. What causes these symptoms? Toxins are secreted by the Salmonella bacteria, which in turn causes the symptoms of vomiting and diarrhea. Give two ways a person with a mild infection of Salmonella can help prevent the spread of the bacteria to other people. A person infected with Salmonella infection can wash hands after using toilet, and the person can also isolate themselves. Okay, the person should not be using any food facilities or handling of food or preparing of food for others. The person should isolate themselves. Alright, the person should also disinfect clothes and surfaces which the person touches, and person must not share utensils, cutleries and towels with an uninfected person. In very serious infection of Salmonella, a doctor can prescribe drugs to kill the bacteria. What type of drug can a doctor prescribe to kill bacteria? Bacteria are killed by a type of drug known as antibiotic. antibiotics. A person with AIDS may take longer than healthy person to recover from a salmonella infection. Explain why. A person who has AIDS, their immune system is compromised. So we can say that immune system of a person with AIDS is weakened or damaged. So it doesn't function properly. The white blood cells within that particular person cannot kill the salmonella as effectively as a healthy person. Salmonella bacteria can be transmitted from chickens to humans. Chickens can be vaccinated to prevent the transmission of Salmonella bacteria to humans. Suggest one other way farmers could prevent the transmission of Salmonella from chickens to humans. The farmer can give chickens antibiotics. In that way, the Salmonella bacteria will be killed. The other ways that the farmer can use is the farmer should not sell the infected chickens that has salmonella, or the farmer should keep the infected chicken isolated, alright, from other chickens that has salmonella. A restaurant owner employed a scientist to test the effectiveness of two chicken cleaning liquids. The scientist took samples from two work surfaces, both the surfaces that has been cleaned with the cleaning liquids. After the surfaces has been cleaned with the cleaning liquids, so the scientist took before and after samples were then analyzed for the number of bacteria they contain. The results are shown in figure 10. We can see with cleaning liquid A in the sink, well, we can see we can see with cleaning liquid B. Now before cleaning and after cleaning, we can see there is a huge difference with cleaning liquid A in both sink and in table, and there is a also a pretty moderate difference with the in the liquid cleaning liquid B. We can see there is a great effectiveness in the table. All right, in both cases it is more. Which cleaning liquid is the more effective one? Give a reason for your answer. So we can choose from here the cleaning liquid B. All right, and because it is more effective in killing the bacteria, because there is a greater reduction in number of bacteria after cleaning in both of the location, in the table and in the sink. The scientists investigated the effect of cleaning liquid A and cleaning liquid B on Salmonella bacteria grown in laboratory. Figure 11 shows the way the investigation was set up. There was a paper disc that was soaked in water, and there was a paper disc that was soaked in liquid B and soaked in liquid A, and Salmonella bacteria was grown on the agar. The petri dish was placed in an incubator at 25 degrees Celsius for 48 hours. After 48 hours, the scientists calculated the area around each paper disc where no bacteria were growing. The results are shown in table 4. With water, the bacteria was growing around the disc, so there was no clear zone. But with liquid A, the clear zone was 11 cm square, and with cleaning liquid B, it was 13 cm square. What measurement would the scientists need to make to calculate the area where no bacteria were growing? The scientists need the radius of the area with no bacteria were growing, and then use the pi r square formula to find out the area. Give one change to the investigation that would allow the scientists to check if the results are repeatable. In order to find whether a particular result is repeatable or not, the scientist must repeat the same experiment and then look to see if the results are similar or nearly similar as previous experiment. The scientists showed the results to the restaurant owner. Both cleaning liquids cost the same per dm cube. Suggest one other factor the restaurant owner should consider when choosing which cleaning liquid to use. Even though both of the cleaning liquids, they are same price, however, one of them has a better effectiveness. So we can take that into account and use the one that is more effective on the type of the bacteria that we're trying to clean. Another thing the particular restaurant owner can take into account is the toxicity of that particular cleaning liquid. If the one that is less toxic is a better choice, the restaurant owner can also take into account the time that is that the cleaning liquid is effective for after the cleaning has been done. Metabolism is the sum of all the chemical reactions in the cells of the body. One metabolic reaction is the formation of lipids. Give one other metabolic reaction in cells. So there can be a range of answer for this particular question. We can say respiration, formation of protein, breakdown of glycogen, or formation of glycogen, breakdown of excess protein into urea, or photosynthesis to form glucose and starch in plants. All of these will be an example of metabolic reaction. So we just need to write only one. I will give you the other points just as a choice. Table 5 shows the mean metabolic rate of humans at different ages. Age in years. Metabolic rate in kilojoule per meter square per hour. So we can see 5 in males, we can see 53, and in 45 we can see in males 36. Similarly, in females at 5 is 53, but at 45 it's only 35. What conclusion can be made from the data in table 5? Okay, so first of all, the first conclusion that we can make is that as the age of males and both females, as it increases, the metabolic rate also decreases. So this one we can conclude. The other one we can conclude is that females generally have a lower metabolic rate compared to male. These are the two conclusions that we can make. However, at age 5, both of them had similar metabolic rate. So let's see the comments now. As age increases, mean metabolic rate of males and female increases. Now we already talked about it. This decreases. So this is wrong. Males have a higher metabolic rate than females after 5 years of age. This is correct. Generally, males have a higher metabolic rate than females. We have already seen that data. Now the next one, the mean metabolic rate of female decreases faster than males up to 25 years of age. Now this does seem, well, we can see the rate is almost similar, alright. However, we can see that females does decrease to 35 really fast, alright, in comparison to that of male, and after decreasing to 35, it doesn't decrease any further up until age of 45. So this might be correct. Alright, let's see the next point. The mean metabolic rate of males and females decreases more quickly after the age of 35. Now we know that for females it doesn't decrease, alright, after the age of 35. There is no relationship between age and mean metabolic rate. Now there is a relationship. So this point is going to be correct. The other ones are wrong because I have explained to you so. Alright, now let's move to the next question. Calculate the percentage decrease in the mean metabolic rate of males between 5 years and 45 years of age. Percentage decrease. Now the percentage decrease will be decrease in metabolic rate divided by original metabolic rate into 100. So if we look into from 5 years to 45 years of age and the rate we are trying to find for the males. So first of all, we will see at age 5 it is 53 and age 45 it is 36. So 53 minus 36 will give us decrease in metabolic rate, and then divided by the original metabolic rate, which is 53 times, and this will give us 32.0754. Now since we have to answer to 3 significant figures, so this answer will be equivalent to 32.1. Regular exercise can increase metabolic rate. Two people did 5 minutes of gentle exercise from rest. Table 6 shows the effect of exercise on their heart rates. Okay, so the person R, it increases up until 99 beats per minute. For person S, it goes to 132 beats per minute. Describe two differences in the response of person R and person S to the exercise. Use the information in table 6. So you can see in both cases, if we look into the data, we will see that there is a general increase in heart rate beats per minute. However, in person A's, the beats per minute at 5 minutes, alright, is 132, whereas person R is 99. That means the person R has, it's more, we can say that the person R is, because the person R is like more active. So the person's R heart rate doesn't increase as fast. The person R heart rate rose more slowly than S. Person R heart rate leveled off, whereas person S heart rate, it continued to increase. As we can see for person R, it levels off at 4 minutes and it keeps that way. However, in person S, it keeps on increasing. And the third comment that we can do for the description is the person R heart rate rose less after 5 minutes than compared to that of S. Complete the line graph in figure 12 for person S. Add the scale to the x-axis and label the x-axis. So the x-axis represents minute, how many minutes have passed. So we can see this one is first, second, third, fourth, and fifth minute. So we will label this as time in minutes. After 5 minutes of exercise, the heart rate of person S was 132 beats per minute. When person S rested, his heart rate decreased steadily at a rate of 12 beats per every minute. Calculate how much time it would take the heart rate of person S to return to resting rate. The resting rate for person S was 78 beats per minute. So it will be 132 minus 78, and since we are at a rate of 12 beats per minute, so it will be divided by 12, which will give us our final answer, and the answer comes 4.5 minutes. A student made the following hypothesis about the heart rate of smokers and non-smokers during exercise. During exercise, the heart rate of smokers increases more than the heart rate of non-smokers. Design an investigation that would allow you to test this hypothesis. To test a hypothesis, we require a group of smokers and a group of non-smokers. So we can take two groups of people, one of them those who are non-smokers and one of them those who are smokers, and then we can have at least 5 to 10 people in each group, and then we will give both of the group person a particular exercise to do. For example, walking in the treadmill at 5 km per hour. We're going to control few variables to make this particular investigation fair. For example, we can use same number of people in each group. That is one way to keep it fair. When using the same number of people, we can keep their gender same. We can give them the same level of activity or exercise. We can choose the people of similar age. We can choose people, and we can, for example, give find out whether they have any health issues or not. So we can take people those who have no health issues and give them same type of exercise and same amount of time to keep this particular controlled variable constant for both of the groups, so that the experiment is fair. And then we can record the heart rate for each person before and after exercise. Then we can calculate the increase in heart rate for each person after exercise and compare the result from each group. Guys, that's all for this particular question paper. Thank you for watching the video. Guys, see you in the next video. Bye bye.